Hello, everyone, and welcome to the Anti-Doping Podcast, brought to you by the Partnership for Clean Competition. I'm your host, Dr. Marie McNeely, and today we are delighted to be joined by our guest, Dr. Andy Hoofnagle. Listeners, Andy is a researcher, professor, board-certified pathologist, and laboratory director at the University of Washington. He is head of the Division of Chemistry, director of chemistry at the UW Medical Center, Montlake, director of Clinical Mass Spectrometry, and director of the Nutrition Obesity Research Center's Analytic Core at the University of Washington. And in addition, Andy is also chair of the PCC's Collagen Turnover Working Group. And listeners, if you missed his first appearance on the show, or if you want a refresher, definitely check out our previous interview in episode 64, where we introduced the PCC's Collagen Turnover Working Group and some of Andy's work developing assays to measure markers of collagen turnover for anti-doping purposes. So today, Andy is joining us in this episode to share some updates on this research and also just talk about the work that he and the group have been doing over the past couple of years. So Andy, welcome back to the show. How are you today?
I'm good. A little tired, but we're good.
Well, we are excited to have you. We appreciate you making time in your schedule. I know this is a busy travel time for you, so we're delighted to have you back. And perhaps just to remind listeners who might have missed your first appearance here, can you briefly introduce yourself and maybe tell us a little bit more about your background?
As you mentioned, I'm a professor of laboratory medicine up here at the University of Washington in Seattle. And before coming to Seattle, I earned my MD and my PhD at the University of Colorado and did my residency here at UW now 20 years ago. After finishing residency, I joined the faculty in 2007 and have been helping to run the clinical laboratories here with a special focus on clinical chemistry, and now help run the clinical chemistry labs throughout the UW medicine system. And on the side, I have a grant-funded research program, which has been funded by NIH and the Partnership for Clean Competition for a long time now.
Fantastic. So Andy, it's wonderful to hear a little bit more about you and your work, and perhaps just to give our listeners a sense of what you do and what kind of research you do in your lab. Can you share what the portfolio of research is that you're working on and how anti-doping science really fits into that picture?
Our lab has been funded by NIH for many years, and it's been grant funded to study cardiovascular disease, diabetes, cancer, nutrition, vitamin D, and kidney disease. We've also been funded by the Partnership for Clean Competition, both through their formal grants program as well as their micro grant program. And our lab really specializes in analytical chemistry. We try to make new assays or blood tests or tests on other body fluids, including urine and cerebrospinal fluid, using a technique called mass spectrometry, which is able to sort molecules by mass, specifically their mass to charge ratio. So using that technology along with liquid chromatography, which can separate the molecules from one another based on things like size and hydrophobicity, or whether or not the molecule likes to be near water or not, we can separate them from each other in time, and then we can use the mass spectrometer to see how much they weigh or what their mass is. So in trying to develop these new assays or tests with an eye towards the clinical lab always, we've tried to bring that technology to the Partnership for Clean Competition, where we've been focused more solely on developing new blood tests to help identify athletes who are using growth hormone, which is really difficult to detect. Our goal is to bring that analytical chemistry and that clinical chemistry to this question of trying to find people who are using growth hormone inappropriately.
Absolutely. And I can appreciate how the methods that you use in your lab can be relevant for so many different fields. And I think it's wonderful that you are applying your expertise to anti-doping. And today we're going to be going in depth on some of your work, specifically on collagen turnover. So to get our listeners all on the same page here, Andy, can you give us a brief overview of what collagen is and what collagen turnover is?
There are actually a bunch of different kinds of collagen in the body. Specific collagens do different things. So type 1, type 3, type 4. Type 4 is really important in the kidney, and there's genetic diseases in type 4 collagen that can lead to pretty serious disease, a range of diseases really. And type 1 collagen is very important for very stiff collagen tissues that we think about, tendons, ligaments, etc. We're going to talk about type 3 collagen today. And that collagen is also found in ligaments and other connective tissues. It also plays a really important role in providing that protective tubing around blood vessels and providing that elasticity that's needed to both maintain blood pressure, but also relax when needed. So collagen is really important as a structural protein in helping to keep the body, all the cells stuck together, as well as our ability to maintain the appearance of being human. But every day that collagen's being remodeled. And whether we're exercising or whether we're growing new muscle or new adipose tissue, which is also called fat, we have to break down the connective tissue and the collagen to allow those cells to divide or to expand or to do whatever they need to do. So as part of that process, we break down the collagen, tearing it apart, ripping it into pieces, and then we build it back up. So we deposit new collagen as we're forming new tissues and new cells. So really, collagen is in this constant equilibrium of being broken down and built back up. And it's tightly regulated. If it gets out of proper regulation, we get things like fibrosis, which can happen in lungs, liver, kidney, and very late stage fibrosis in the liver is called cirrhosis, which I think people are pretty familiar with. So this kind of tight regulation of collagen and its regulation at the hormonal level is really what allows us to understand why growth hormone could have an effect on collagen turnover, since collagen turnover is partially regulated by growth hormone. So just to summarize, collagen is really important for holding us together. We constantly remodel it because our bodies are constantly changing, and it's very tightly regulated. And so it should be relatively constant and equilibrium in every person over time.
That makes sense. And that was a helpful explanation. And then to dive in a little bit deeper, you mentioned we're focusing specifically on the type 3 collagen. How is turnover of this type 3 collagen relevant then specifically for anti-doping?
This really goes straight to growth hormone and growth hormone abuse in athletics, in particular in elite athletes. By injecting growth hormone exogenously, we can potentially tilt the collagen turnover into more of an increase in mass and increase in performance. The regulation of the collagen turnover is in a way indirect in that growth hormone can actually increase the production of IGF-1 and lead to remodeling. There have been some studies that suggest that this exogenous injection of growth hormone is good for performance. So now many people are using it. Many athletes are using it in the hopes that their performance will improve. But it's important. There's a lot of other studies that suggest that it doesn't do anything at all. And perhaps it's important in combination with other doping approaches. But to really put it out there, we also know that patients who have very high concentrations of growth hormone have bad outcomes. They have acromegaly, which is an enlargement of many of the larger bones in the body and can lead to cardiomegaly, which is an inability of the heart to pump blood properly. We know that too much growth hormone can cause serious heart problems. There is some evidence, it's not very strong, that growth hormone can improve performance, but we're pretty confident as an anti-doping field that a lot of athletes are abusing growth hormone in hopes of improving their performance. And being able to understand how growth hormone is regulating collagen turnover and can we detect aberrations in collagen turnover after people have injected growth hormone. The hope is that we protect the athletes from what could be the deleterious effects of growth hormone, as well as the kids that look up to them.
Definitely. And I think biomarkers can really help maybe give a window into what is going on in this process of collagen turnover. So, Andy, can you explain what are some of the potential biomarkers of collagen turnover that you've been looking at?
So procollagen is a pretty large molecule. It's not the biggest protein in the body, but it's pretty big. So it's made up of a fibril, which is the collagen molecule twisted up upon itself. There are 3 molecules twisted up into a twisted strand. And on either end of the molecule, there are propeptides, both on the one end of the molecule, which is called the amino terminus, and one on the other end of the molecule, which is called the carboxy terminus. And as the collagen is being released from the fibroblasts, it comes together to form that helix, that twisted strand of 3. And on the one end, the amino terminal propeptide of procollagen type 3 can be clipped off. We call that P3NP. And on the other end of the molecule, the carboxy terminal propeptide of procollagen type 3 can be clipped off. That's called P3CP. So those are both markers of collagen deposition. As collagen's being laid down, those are clipped off as part of the process of making that mature collagen helix and then bundle. There are other peptides that can be released as the collagen is broken down from the main collagen fibril itself, and we call those degradation products. Other things called telopeptides. We're not going to talk about those today. What we're really focused on is the markers of collagen deposition, P3CP and P3NP. as well as the degradation products that are being released as that collagen is being remodeled. So the hypothesis on what we're working with the collagen turnover working group to show is that multiple biomarkers within the pro-collagen molecule could give us a better window into the abuse of growth hormone in athletes.
Definitely. And as you alluded to earlier, it is difficult to identify growth hormone abuse in athletes. So, Andy, can you explain the two different approaches that are currently approved by the World Anti-Doping Agency, or WADA, to detect growth hormone abuse?
The first is the direct detection of growth hormone isoforms. And when the pituitary gland secretes growth hormone, pituitary gland is a gland in our brain. And when the anterior pituitary gland releases the growth hormone, it has different isoforms that are in a relatively constant ratio from person to person. We can only inject a specific isoform, which can tilt the relationship of those isoforms away from what we see endogenously to enrich for that injected form. When we inject growth hormone, we do get suppression of growth hormone from the pituitary gland itself. So if we're able to detect a change in the relationship of those isoforms, it's a direct detection of the injected hormone itself. Unfortunately, the half-life of growth hormone is very short. So someone who injects growth hormone the night before and then gets tested the morning after, we're unable to detect the growth hormone itself. Sometimes we can detect, again, an aberration in the ratio. But since it's such a short half-life and since the effect on that ratio is short-lived, it's very difficult to detect. The second way that we can detect growth hormone is the result of the downstream effects of growth hormone. So the two, we call them biomarkers. They are proteins in the blood circulation that we can measure the concentration of to try to figure out if someone did use growth hormone recently or not. And those two proteins are IGF-1, or insulin-like growth factor 1, and P3MP, that fragment of type 3 procollagen that we talked about just a moment ago. The problem is with what we call this biomarker strategy, or looking at the downstream effects of growth hormone, is that in each person, there will be a different baseline level of IGF-1 and P3MP, and the change that each person will see when they have exogenous growth hormone injected into them will be a little bit different. So we really want the smoking gun, but it's hard because everybody's a little bit different. The way that the anti-doping field has dealt with this is to look longitudinally and look at the concentration of IGF-1 and P3MP in people over time to look for evidence that there are rises in those two biomarkers that would be indicative of using growth hormone and abusing growth hormone in sport. So we're looking for an increase over the baseline as suspicion for the use of growth hormone. So two pretty different ways, either direct detection or indirect detection, that someone has abused growth hormone.
That makes sense. And if people are seeing this increased pattern over time, or maybe a spike in the biomarkers that they're looking at, maybe in that athlete biological passport, for example, are there other confounding factors that could explain it?
Unfortunately, yes. There is some evidence that in some people, biomarkers will go up if they are recuperating from an injury, for instance, which is also, unfortunately, the time that people may abuse growth hormone as they're trying to recuperate faster. So that can be a potential confounder. There are others, but that's probably the most important one relevant to this conversation.
That makes sense. And then thinking about the collagen turnover rates as a different approach, a third approach that people could consider, what are some of the benefits and limitation of this approach to identify growth hormone misuse versus some of these other available approaches?
I think it all comes down to sensitivity and specificity. We know that IGF-1 and P3MP by themselves are variably specific and variably sensitive. And we're hoping that collagen turnover, again, combined with IGF-1, will be sensitive enough to detect people who are using growth hormone. That's really the question that we still need to ask as a turnover working group. As part of that, we have to really be accurate in the quantification of those different pieces and parts of the protein. So we want to be sure that when we run the assay, whether it's in Salt Lake City or London or Los Angeles or Cologne, that we get the same answer regardless of who's running the test. We know that immunoassays have problems with that. We published back in 2014 a big issue with the Siemens assay for IGF-1. It was really taken off the market after our publication. But the most important thing to remember is that the technique of mass spectrometry, which has already been deployed in all of the WADA labs, that technology can be used to quantify proteins. It's already being used to quantify IGF-1. If we can leverage that to do a better job quantifying P3NP and these other pieces of pro-collagen type 3. The hope is that we'll be more sensitive and more specific in quantifying actual collagen turnover. There are lots of proteins. I mentioned that there are many different collagens in humans. We need to make sure that we're specific. That's another problem that immunoassays have. It's not just variability between labs and between days, et cetera. But sometimes they're picking up proteins that aren't the protein that they meant to pick up. And in detecting related proteins, that lack of specificity can lead to some confusing interpretations. So we have work to do. We know that we're accurate. We know that we're sensitive. We know we're precise. But what we're lacking is to know if it's clinically sensitive. I'm using that word to borrow from lab medicine. We need to be sure that our signal is tied to the actual event that we're trying to detect. And that's the work that needs to continue.
Well, that makes sense. And I know the last time we spoke was all the way back in 2021, which feels like eons ago. I know there have been some exciting updates since then. Specifically in 2022, you and colleagues published a paper that described methods that allow you to simultaneously quantify multiple different biomarkers of collagen turnover. So Andy, can you talk a little bit more about this method that you developed and why it's so significant?
We've, in our laboratory, been trying for a long time to quantify low abundance proteins in biofluids, and efforts at trying to quantify P3NP had been coming up short for a long time. We, in trying to quantify procollagen type 3, had identified multiple peptides from procollagen. We couldn't ever identify a good peptide from P3NP, which I'll get back to. But that original paper showed that we could use antibodies as reagents to grab on to peptides after the proteins had been chopped up into smaller pieces called peptides by a protease called trypsin. So we can use trypsin, digest all the proteins up into peptides, and then use an antibody to grab onto the peptide before analyzing it with liquid chromatography and mass spectrometry. Those antibodies were key to making the assay as sensitive as it is. And the mass spectrometry was key in the specificity of the method. So the paper that we published now a few years ago demonstrated that we could quantify these low abundance biomarkers, these pieces and parts of procollagen type 3, specifically the P3CP and the degradation products. So it was a great advance in being able to quantify procollagen, but we're still missing a piece of the puzzle, which was the P3NP. One of the tricks that Huu Hien, who is a junior faculty that works with me, he was a postdoctoral fellow at the time, but he and others in my laboratory identified a trick that would allow the antibody to bind even more tightly to the peptides, which also improved the sensitivity. So it was an advance both for the quantification of peptides from procollagen type 3, but also as an advance in the field to help improve the sensitivity of other assays.
So let's maybe talk a little bit more about, in detail, the results that you saw and perhaps the potential impact specifically for anti-doping and how these results could be applied.
I think the most exciting advance for anti-doping is that specificity, similar to what we had done with IGF-1 in removing the immunoassay and all of its potential problems from the measurement of IGF-1. We can now do that for procollagen fragments. And so by having a specific assay, we have a lot more confidence in the concentrations that we're measuring and a lot more confidence in trying to identify people who may be misusing growth hormone.
Absolutely. And Andy, I know a moment ago you mentioned that at the time you were not able to get a good peptide from P3NP. So perhaps we could dive into this mystery a bit more. Can you tell us a little bit more about what the story is here?
Well, our new paper that just came out, the reason it was such a fun paper for us is that it was a collaboration of everybody who is on the collagen turnover working group. Danielle Montlake, who is a former PCC fellow, did some work a few years ago to identify the peptide that we could never find. And we couldn't find it because it was actually modified post-translationally, which identified that slightly different form of the peptide. And we were able to make an antibody to it. And when we did, we got really amazing sensitivity for P3NP to add to exactly the same assay. So we're measuring all these peptides at exactly the same time using different antibodies to pull down the different peptides at the same time. The mass spec can tell the difference. And so that was a very exciting advance and really made possible by being a collaborative group in the collagen turnover working group. So now we have 3 different antibodies. One is targeted to the P3NP, one's targeted to P3CP peptides in each of those, and then the degradation products that come from the collagen fiber itself. That was exciting. The other thing that we did was we moved it from individual Eppendorf files to a 96-well plate. What that means is that instead of someone being able to process somewhere between 12 and 24 samples at a time, we could ramp it all the way up to 96. Not quite 96 because we have to have calibrators and quality control materials on each of the plate, but we can do a lot of samples in a single batch. That means that anti-doping laboratories can measure dozens of samples in a single day, which makes it a lot more feasible to replace the immunoassay, which in many cases are on automated analyzers. You just sort of walk up, put the tube on, press a button, and it happens. We're not there yet with our technology, but at least if we walk up and someone spends 4 to 8 hours in a day, they can get back many, many, many results, which is exciting. So we've had a lot of success in the collagen turnover working group, collaborating together and coming up with now an assay that we're ready to share between laboratories, which is ongoing.
I think that's fantastic. And these results were recently published in a paper that listeners can go out and find online. And this is a high throughput assay, as you mentioned, being able to switch over to that 96-well plate for a variety of different collagen turnover biomarkers. Can you talk a little bit about the reliability and validity of this method?
Those are loaded terms. I appreciate them. The most important characteristics of a biomarker or any blood test really is precision and linearity. Precision in that we can get the same result day after day, month after month, laboratory to laboratory over years. And that's what sets clinical chemistry apart from many other analytical chemistry laboratories where they really only have to be successful for a week or a month to get a project done and then they kind of forget about it. In clinical labs and in anti-doping labs, our goal is to be able to do the same thing we're doing today 10 years from now and still get the same results. So we try to do as many validation studies as we can to demonstrate that the assay performs as well as we expect it to. So we have criteria that we expect the assay to meet during these validation studies. And what I can say about this 96-well plate assay that the working group developed is the assay is precise and we can get really reliable results over months in our laboratory. And again, what we hope to prove is that we can get it in different laboratories around the world. We also demonstrated that it's linear. When we put in twice as many protein molecules into a sample, we want to make sure that we are able to say with confidence that there's twice as many molecules in the sample. So with careful calibration and thinking about the different kinds of interferences that we might have in samples from humans, we were able to demonstrate linearity in the presence of many potentially interfering substances. So yeah, this moving to a plate and demonstrating precision, linearity, stability, and freedom from interferences gives us a lot of confidence that the method is reliable. And I think we validated it well enough in our lab that it's worth trying to get other labs to be successful as well.
Absolutely. And I think the increased throughput is a huge benefit. Does this also translate to requiring less sample per test or per well than you would with the larger tubes that you had?
Huu Hien did a lot of work to try to see how small of a specimen volume we could go. The original assay used 400 microliters, which isn't a ton, and way less than a teaspoon or a tenth of a teaspoon. But now he got all the way down to 100 microliters for the plate assay with good sensitivity for all three. And we're trying to get even smaller so that the amount of blood that we would have to take from an athlete can be minimized even further. So yeah, the transition from tube to plate increased the throughput, but it also reduced the specimen volume while maintaining the sensitivity that we need, the measurement of all the peptides involved in collagen turnover.
I think that's remarkable. And I think this new approach also, you worked with capillary blood samples that were collected using the Tasso Plus microsampling devices. Listeners might be familiar with this device from some of our previous episodes. So Andy, can you comment on why capillary blood versus venous blood compatibility was important in developing this assay?
It sounds like you've done other shows and thought out loud about capillary blood and how it could really transform not just the anti-doping industry, but the clinical laboratory testing industry as well. It's really hard to get people to come in for a blood draw when they live tens or hundreds of miles away from their blood draw facility. It would be amazing if they could use a device like the Tasso or Tasso Plus or one of the dried specimens that they're able to generate. It would be amazing if people could use the device to draw their blood, put the cap on it, put it into a box and send it to a central laboratory for analysis so they don't have to leave their home. It has implications for diabetes, kidney disease, all sorts of chronic monitoring of drugs and therapeutic drug monitoring. It would be a game changer. But when we draw blood from the capillaries, the blood is not the same as sticking a needle in a large vein. And we're still sorting that out with respect to collagen turnover. There was a good agreement or fair agreement between what we were able to see when we draw venous blood and analyze the serum versus the Tasso Plus device. But the correlation or the agreement between those was not as amazing as I would like. And we're still trying to figure out exactly why. But if we could use the Tasso device and we could measure IGF-1 and P3MP in the same sample or other proteins if they become important and available, that would really change our ability to do the biological passport in a meaningful way, where people don't have to disrupt their training and the rest of their lives to get their blood tests. They don't have to have a phlebotomist go and track them down to draw their blood. We can really either video someone while they're doing their Tasso device or some other way to have the athlete be able to do it while they're in the field, whether that's in the mountains or the track.
That makes sense. I think there's a lot of potential just thinking about anti-doping broadly with capillary blood, and particularly sports that may involve people's hands where they don't want to do a finger prick. So having just the shoulder collection, or they're using their arms a lot, they don't want to have the venipuncture. So I think there's a lot of potential there. And we mentioned, of course, Huu Hien, who is a former PCC Research Fellow. So I'd love to talk about your experience working with him and just being involved in this PCC Research Fellowship experience. So can you talk about what it's been like to be a mentor for a PCC Research Fellow?
Huu Hien is an incredible scientist. He's brilliant, innovative, motivated, incredibly curious. And it was really fun to be able to bring him into our laboratory and let him thrive. He wasn't just involved in the research that we did. He really led it. He really drove the science in many ways. And that's really inspiring. It's fun to work with him and the next generation of scientists and watch them go. I'll be honest that probably sounds cliche, but I learned as much from him as he does from me. He was a metrologist before he came, and metrologist is the study of the measurement of things. So he spent many years thinking about how to get the most accurate result using the analytical chemistry techniques that he had learned over the years. He brought that knowledge to our laboratory and really put it in place, taught all the rest of the people in my laboratory how to do it. So I was able to mentor a really talented scientist who could learn a new field and apply all of his amazing knowledge to that field and think about taking it further. So it's inspirational. It helps me think about how I can learn from others nearby in my own laboratory, and then to take that, again, collaborative effort and run with it. It's been incredibly rewarding and inspirational for me.
Definitely. And I think maybe taking a step back to think about the bigger picture, Andy, what do you see as the potential impacts of this PCC fellowship program on the field?
Well, it brings new people to the field, people that are either still in younger parts of their training all the way up through someone like Huu Hien who has lots of experience. Danielle, who found the peptide for us, was using technologies that she had learned during her graduate school and applying it to a new problem. It's really that infusion of new innovative ideas that I think the fellowship program can bring to the field of anti-doping, which we are trying to stay at the cutting edge of the science because we're trying to stop a practice that's continually evolving in some respects to try to evade detection, but also new ways to try to cheat the system. So I think it's great to have new eyes, new brains, and a fresh perspective on a problem that's really been plaguing athletics for a long time.
Definitely. And I know you, Huu Hien, and the whole Collagen Turnover Working Group have made some important strides in these questions related to growth hormone. But what do you see as some of the biggest challenges that remain, Andy, in the problem of detecting growth hormone abuse and maybe finalizing or implementing these collagen turnover assays for routine use in all the labs around the world?
It's interesting that you asked that. We've had a recent development in the United States where Siemens has bundled their P3MP assay with two other biomarkers in a liver fibrosis panel. And in so doing has increased the cost of doing testing for P3MP fourfold, which really makes it financially, I would say, unviable. In addition, they actually deprecated one of the instruments where they used to be able to run the P3MP assay. So now one of the anti-doping labs in the United States can't even run the assay. Even if they wanted to spend 4 times more for it, they can't run it. So there's actually a pretty important imperative to find a solution. I think this LCMS approach, whether it's for P3MP or for all of the collagen turnover biomarkers, we have our marching orders as a working group. The hurdles, though, what's potentially in the way, we really do have to prove that the assay that we have developed can be tied to the activity of using growth hormone. So we did a great job working with IGF-1 in the past. We've shown collaboratively as part of the PCC, we can develop technology, deploy it, and then have a lot of labs around the world use that technology. We need to do that again. We need to prove that the assay can be run in other laboratories. It is a multi-step process. The standard operating procedure is many pages long, but I think that we can do it. We have some really good preliminary data, and I'm really hopeful that as a working group, we can pull it off. All of those inter-laboratory studies, comparisons, deployments are underway. So hopefully the next time that you and I chat, we can share a success story.
That would be wonderful. And you mentioned this quote unquote clinical relevance and making sure that you're able to tie what you're measuring to the actual activity of taking exogenous growth hormone. How do you solve that problem or start to answer that question?
Working with USADA and PCC, it's our goal to get specimens from athletes that have been collected over many years. And we can begin to build a distribution of what we expect within people as well as between people so that we can get something akin to the growth hormone 2000s, which is currently used in the biological passport to monitor over time. We need to be able to demonstrate that the distribution is as we would expect it to be in a normal population of athletes. They also have, in collaboration with Smyrtle, the Sports Medicine Research and Testing Laboratory in Salt Lake City, they have a pretty active program of administering growth hormone to individuals and then collecting blood specimens during many days of injection and then following. And we can use those samples to look at the results from our assays and demonstrate that, in fact, we can detect that rise in collagen turnover biomarkers or the relationship between those biomarkers as a marker of growth hormone administration. So again, we have work to do. The first step is to show that all the labs can get the same results and then to split these samples up between each of the laboratories so that we can begin to understand the distribution of normal as well as what the deviation from normal.
It sounds like you and the group there have a lot of work to do to keep you busy. And I know the PCC was thrilled to have you as a presenter at the recent conference there in New York City. So Andy, I'd love to talk a little bit about your conference experience and what was it like presenting with the group and meeting everybody there?
It's such a fun conference because it's a mix of so many different kinds of people. There's athletes, there's scientists, there's regulators, there's sports leagues. I met one of the horse racing testing groups. It's just a really wonderful group of people that, again, bring very different perspectives to the same problem. Everybody's hoping to do right by our athletes, our competitors, our national representatives. I'm a broken record. I found it inspiring. I'll use that word again. So there's that feeling in the room that we can help, that we can do something to protect athletes, protect the fairness in sport, but also all the kids. I mentioned them before. There are kids who look up to these athletes and just national champions. And I want those kids to know that they don't need to use growth hormone to do what they did.
Definitely. And I think it's just so valuable to get everybody in the same room to have these conversations and make these connections between different parts of the wonderful field of anti-doping here. So if our listeners are inspired and want to learn more about you and the work that you do, Andy, what is the best way for them to do so?
The PCC newsletter actually puts out a summary of what the Collagen Working Group is doing every couple of months or every 3 months. So I think that's a great resource just to kind of keep tabs on what we're doing. For my own work, there's webinars at an organization called MSACL, which is Mass Spectrometry and Advances in the Clinical Laboratory. And I've given a few talks that talk about the technology at its foundation, sort of what is the scientific underpinnings. All the papers that we talked about today are available on PubMed. I would encourage people who want to know enough about the science so that they can make recommendations to WADA or to their own local anti-doping associations. We tried to do a really thorough job explaining how we developed the assay and then a standard operating procedure that is incredibly detailed so that anybody could run the assay in their own laboratory if they wanted to. I will say that I'm not on social media. I'm too old. I'd have to rely on my daughters. one of whom dances 5 days a week and the other one who plays competitive soccer 5 days a week. They're in middle school and high school, and they spend a lot of time using their bodies in amazing ways. And so
cool.
Maybe they can teach me with their amazing brains how to use social media properly. But that's not a way that I connect with people.
It's never too late to learn, Andy. You've got this.
Right.
Well, thank you so much for sharing those resources. Listeners, definitely take some time to check them out. And Andy, it's been such a pleasure to have you on the show today. Thank you again so much for your time.
Thank you, Marie. I enjoyed it.
Well, it's been wonderful to chat. Listeners, we hope you enjoyed the conversation as well. And if you haven't already, please take a moment to rate our podcast and leave a review on your favorite podcast platform. And listeners, please note that the Anti-Doping Podcast features a wide variety of clean sport experts, and episodes may include statements and/or viewpoints that do not necessarily reflect the views or values of the PCC. Thank you, and we look forward to seeing you on the next episode of the Anti-Doping Podcast.